how complicated it is, you can understand why some microbes that are getting the absolute sweet life may decide not to do this because it is fairly complicated and lots of resources involved. They have to be well fed and everything going good in order to do that. And then when you think about the craziness of some microbes doing multiple flagella for each one of their, I mean, it's crazy, especially Paratrichus, where they have a bunch of flagella all inserted, so to speak, around their entire cell. So it's rather amazing. So we've looked at that and how it is a way for them to move in a directed manner from point A to point B. We will look at movement, motility in lab this week or next week. So we'll come back to this idea, at least in the lab, how to identify it a little bit better. But the fancy flagella isn't the only way for microbes to move. There are some other options. And gliding motility, they don't know as much about this compared to the flagella. So there's still some murkiness on all of this, but I think we can say some things with pretty good certainty. It doesn't seem to involve the flagella. That's not what's going on here. So it, under the microscope, or if you video it and really look at it and study it, it is slower and smoother. Not so, like I said the other week, microbes that have flagella, especially if they just have one flagella, they tend to be kind of herky-jerky and they look like they're really hepped up on caffeine. So you don't have that going on. It's much slower, and it's usually a rod-shaped cell, not always, but usually. And so you see the movement occurring along their long axis, along the long side of their rod shape. And they've done studies where this kind of motility does not work if they're suspended in water. They have to be making contact with something, whether it's the outer surface, a rock, a log, the soil. They have to have some kind of physical contact in order to get this to work. And I'll show you a little bit why they think that. And so some microbes that tend to do this, they seem to have evolved this ability to just kind of excrete this polysaccharide slime that helps them move. It's similar to the idea of capsules that you studied in when you looked at capsule slides in lab, and I'll talk about capsules in here too. It's kind of the same polysaccharide slime, but they're actually using it to move instead of just protecting themselves. Then there's some bacteria that have done a different mechanism. They use pili, and they've actually named them type 4 pili is what they call it. So these are pili that actually will be used for movement. And when they look at it and study it, you know, pili are long and skinny, kind of look like hairs coming off the cell, but they're hollow. Protein, it's just protein subunits. And what happens is the pilus will like reach out in front of the cell and kind of dig its tip in to the solid surface and then kind of ratchet its way forward and just keep doing that. It's a really bizarre adaptation, but it works. It's kind of like, what's that thing that you put over the grappling hook? Yes, grappling hook. It's kind of like the grappling hook idea where you use the grappling hook to hook in and then you can climb and then there's some bacteria that have in their cell membrane and cell wall area along the long axis, they have these proteins embedded that seem to really only be there to help them move. And it's almost like, I kind of think of a tank treads on a, army tank. That's kind of what I think of, except it's not really rotating per se. But it's that kind of action where the proteins make contact and then just kind of ratchet the cell forward, pushing the cell forward. Speaking of tank treads. I drove by Presque Isle and there's that bike there, you can rent bikes. Have you seen those? They're like the back is a tank tread and the front is a ski. I drove by kind of quickly. I wasn't speeding, but it caught out of my eye. I'm like, corner of my eye. I'm like, what is it? Does anyone know about that? Tried them?
Style they're renting them?
I can't remember the name of the business. It's right on Presque Isle, just somewhere near the Wooden Nickel. Not that I know about the Wooden Nickel per se, but it was in that area. They had a bunch of bikes out and the headlights were on. They had a big headlight. But it was just like a tank tread for the back and a ski for the front. But it was a bike, a pedal, like a, a pedal bike you get on. Has anyone tried that? I was wondering how much they rent. I'm assuming they rent them because there's several of them lined up. Anyways, check that out if you want something fun to do in the winter. If it's not too expensive. So anyways, here is that tank tread idea where they have these specialized proteins embedded in the cell membrane and the cell wall that will make physical contact with the, some solid surface or semi-solid surface and allow it to kind of move forward, push itself forward. And so it's still, they don't really understand it too well. They think there needs to be some energy expended, so proton gradients. I'm not going to ask about that. Because this method in particular is not well studied, but basically understanding some of the options here that they have is a good idea, but that's about as deep as I'm taking it. Here are some images of, on the left, you can see they are motile. They're on an agar surface and they spot inoculated the bacteria and then incubated the plate, and you can see how it's moved out from that initial spot. And then these are colonies of the same bacteria that was made a mutant for being motile. And you can see that the colony just stayed localized. It didn't have any of the cells moving off. So if you see, oh, here's another one. This is a soil dilution that I did and plated out dilute sample of the soil. And you see a couple of things on here. You see all this crazy hair-like growth. That's probably a bacillus species. That's the genus name Bacillus. And could be Bacillus circulans. There's another one though, which I'm blanking, that can also look like this on an agar plate. So that's motility. They are moving around. And then if you look up into the very top portion, I don't know if you can see what's going on here. I'm going to blow that up in the next slide. So you can actually see this kind of slime layer from the colonies. And so that's another type of motility, probably the polysaccharide type. And so you can actually see that on some Petri plates when you isolate bacteria, especially from their natural habitats, that they'll be motile like that.
So the whole colony is moving?
Well, that's what I looked into it a little bit, and that's what they're showing in at least the things I was reading up on. They said that could be what's going on. And there's this phenomenon called swimming and swarming. And so bacteria can essentially live two lifestyles, the swimming lifestyle and the swarming lifestyle. This is swarming. Swarming means they all gather together as a team, and then they all move across the surface as a big unit. So they think that's what's going on here. At least that's what I was interpreting when I was looking for answers to that. And so again, that solid surface. So if they were starting, let's say this, colony started way back here and then it moved and now it's over here. Why do you think it moved? What's it trying to do with that? More nutrients, yeah. So when you played out you did the streak plate and the spread plate, and then you came back after incubation and you looked at your plates and you had colonies, hopefully individual colonies. Did you get individual colonies on some of your spread plates that you could count? So you have to appreciate that colony that we see with our eye, that's millions of cells. all clustered together, originating from that one cell that you put there when you did your streaker spread plate. Well, after a while, all the nutrients right around that are gonna go. And then they're like, well, now we're starving, now what do we do? If you're modal, by some means, you can start to, in this case, swarm. And it's actually almost like a predator type relationship that they form with each other in the sense that they team up, like, kind of like wolves in hunting packs. And then they'll hunt together, so to speak, looking for other nutrients. And if it was, patch of soil, they'd be looking for things they could attack, essentially other living cells that they could attack and drain them of their nutrients. So you have some really interesting designs on auger plates. Here's one that they did, and then they colored it to make it look pretty. But you know that they what they did was they put the bacteria here and then incubated the plate. You can see over time how much it went hunting for nutrients. It really went all out. So you can get these types of patterns. What you do is you make an auger recipe that's really low in nutrients. Or if you get really fancy, you can try to create a gradient of nutrients so that you can get some interesting patterns as they hunt for their nutrients, so to speak. So gliding motility is several different mechanisms. It depends on the microbe and what kind of habitat they're adapted to. It's not as fast as flagella, and they need a solid surface, but it can work. All right, here's a question for any of you who are scuba divers. If you've done scuba diving, it took both the intro and advanced scuba class here when I was an undergrad, which is pretty wild. First time you breathe underwater. I don't know if they still do it where they take you into the lap pool and you just sit on the bottom and breathe. That was amazing to me. But anyone take advanced scuba here? Well, be prepared to get cold. Because, oh my goodness, every dive, at least when I took it, every dive in the advanced scuba was outside. No matter, so I was diving out in December. I took it in the fall semester. So that the month of December and November, it was cold. But anyways, you learn about the idea that, you know, you certainly have flippers that you can use to swim around. But do you know how, kind of an easier way to control your, like, where you are in the water column? It's not really being shown there, but you have your BCD, they call it, the buoyancy control device, and it's actually, you can pump air into it. So it's like a fancy life preserver in a way, but you can, you know, so you wear weights, you have a weight belt, you have flippers, but that's a lot of energy to keep moving your feet to stay steady in a column of water where, I just want to go 20 feet and stay there. So you use your VCD to inflate, and between your weights and the VCD with air pressure in there, you can just stay stable at 20 feet and not sink or float. And so it's pretty fun. We did a night dive right at Black Rocks, the cove area there where that's where we did our night dive, and we all got in there and sunk to the bottom, and then we all turned our lights off. And it was just pitch black, and you could hear people breathing, you could see the bubbles. Lots of fun if you ever get a chance. It's not like the coral reefs. Obviously, we don't have the coral reefs here, but if you want to experience something truly amazing, I was very happy that I had that experience. So that's the idea of what we see here, gas vesicles. Instead of a BCD, they have gas vesicles. So it's kind of the same thing, at least in theory. They're cells, and inside the cell, they have these little compartments that they can fill with air and deflate. Not really deflate per se, because it's fairly rigid, but they can get rid of some air and put some air in. And so what you're seeing here is Lake Mendota, right on the University of Wisconsin-Madison's campus. They have a nice, like their northern, our northern center, they have a union center or whatever they call it, where people can go eat and drink beer. And it's right on the lake there. And sometimes at the height of summer, you'll see all that green and during the day like that. And these are aquatic bacteria. Why do you think they might be green? Yes, they're photosynthesizing. So if you have that capability to utilize light energy to get your ATP and sugar made, I mean, that's an awesome ability. If you have that ability, you'll take advantage of the sun. When it's daylight, it makes a lot of sense that you'll be right up at the surface and absorbing as much sunlight as you can. But then during the night, you can't do photosynthesis. So then you might go, I'm a little exposed here. All kinds of fish and birds, and I think I'm gonna sink. And so during the night, you sink down and try to kind of stay away from other things that might eat you, attack you. And so being photosynthetic, chances are if you're aquatic and you're photosynthetic, you'll have some kind of gas vesicles so that you can move up and down in the water column and strategize so that you can do your photosynthesis and try not to get hunted and eaten. And so all of these kind of bright yellow areas are the gas vesicles. Here you're seeing an electron microscope picture and all the light gray. So some of these look, this is if you look on end, and this is if they make the cut across the surface. So it's kind of a longitudinal BCD or rod shape, I should say, BCD. And this one, you're seeing just different viewpoints, whoever took the section. So, but the upshot is here, you see a lot of these vesicles inside the cell, and all of those can get manipulated with how much air is in there. So pretty simple mechanism, pretty, I mean, it doesn't really cost you any energy or anything. It's just air in, air out, and it gets you there in the water column. Okay. And then you don't have to write this down because it's in the PowerPoint. I'm just going to list these out really quickly. So these are different ways that microbes will move in response to their environment, and they're all different taxes. Chemotaxis with chemicals, both chemicals they want or chemicals they want to avoid. Phototaxis, again, if you're photosynthetic, you want to move toward the light. Aerotaxis, if you like oxygen, you might want to go toward it. If you hate oxygen, you might want to go away. Osmotaxis has to do with how many solutes are dissolved in the solution around you. And some bacteria respond really well to that. And then hydrotaxis is looking for water. So, I mean, most of these make sense if you just pick apart the word and look at it. But basically, what we're trying to get you to think about here is that microbes are very sensitive to their environment. You know, minute by minute, they're assessing their environment so that they can get their needs met. And even though they're single-celled and we can't see them with the naked eye, they are very, very busy and active and are pretty sophisticated at sensing and then responding appropriately to their environment. So here's this chemotaxis idea where you can do this under the microscope with just, put a microscope slide down, put maybe a sugar solution in one edge of the microscope slide, and then you put your microbes down and maybe a cover slip. So it's allow them to move around a little bit, and you'll see that they'll start to sense this attractant. And, of course, if you put a sugar solution on one edge, it's going to start to diffuse through that water on the slide, and they start to sense when they start to get into more concentrated areas, then they get a more directed movement if they are modal. You can see that they'll start to orient to the sugar solution in this case and move toward it in a more directed way. And so we actually refer to when microbes are moving, when we're looking at, especially under the microscope, you will see a run period. And that's when they're making solid progress from point A to point B. And then there'll be a tumble period where they will stop and kind of reassess what's going on. And if they need to change direction, they can do that. And then they'll go for another run. And so run and tumbles as they move. In this case, they're moving toward an attractant. And you can do all kinds of experiments with this type of ability for them to sense and respond. You can do attractants, you can do repellencies. So these are like micro capillary tubes you put into a little beaker of water with the microbes growing in there, or maybe a broth solution or something with microbes. And you can see that they'll go up into, in this case, they put a capillary tube that has like a sugar solution or something. Whereas maybe this is an antibiotic and you can see them moving away from it. So they're constantly assessing like that. And if they are modal, then they have this great opportunity to behave appropriately and hopefully survive longer. This picture here, that white blob in the middle, that's oxygen. And you can see that they color coded a bunch of the different bacteria, but you can see them really starting to congregate around the oxygen. So these are the oxygen loving bacteria that are like, yes, I want that oxygen. And so they're starting to cluster around that. So all kinds of experiments showing, you know, that microbes can move and behave appropriately. The software. I don't know if it says it in the notes. I might have it in the notes section. Let me just see. I don't know if that picture is from your book or not. They might explain a little bit more. So it says tracks of modal bacteria in seawater swarming around an algal cell, taken using a tracking video camera system attached to a microscope. They're responding positively to oxygen produced through photosynthesis by the algae. So yeah, they did a video camera attached to the microscope, and then they color coded they probably labeled the bacteria with fluorescent proteins so that they could, you know, pop against the background like that. We have a confocal microscope in our department that would allow kind of that idea too, where you can keep the organisms alive as you study them under the microscope and get some really nice images like that. So microbes can, you know, many microbes have ways to move, not all though. There's plenty of the microbes that you'll work with in lab, they can't move. And so when we run the motility test, hopefully the ones that are supposed to move will move for you. And we'll have flagella. Usually we work with the ones that have flagella. So hopefully that experiment will work correctly. All right. What are some other structures that you can see sometimes if you do proper microscopy and staining? Here's another idea. Fimbriae, that's plural. Fimbriae would be singular. Fimbriae. Again, these are long, well, not super long, but longish strands of protein subunits put together. And they are not as long as pili or flagella, but they usually are more numerous. So if a cell decides to make fimbriae, it's usually a lot that's just peppering the whole surface of the cell. And when they look at what it's doing, like why would a bacteria want to do that? What kind of advantage? Mostly it's attachment. A lot of just really solid attachment, especially if you have a lot of fimbriae, it really does help you to attach. And so it's fairly typical that pathogens that attack other organisms, whether it's a human or another animal, or even a plant, they tend to have ways to attach. And so fimbriae is a good strategy because they're not as sophisticated as flagella, so they're easier to make. And if you make a lot of them taken all together, that's a pretty good contact force that you can really stick to the surface of whatever. They can also be involved in helping them to stick into the biofilm. I'll talk about biofilm a little bit more too. But just helping them to attach and to some kind of surface. Here's a picture where you can see the longer, much longer flagella and then the fimbriae. We've already talked about pili. So again, they're longer than fimbriae. Protein subunits that make them, and they're long and hollow. We've already talked about the sex pilus, so to speak, that's done during conjugation. What's getting transferred to the sex pilus in conjugation? Plasmid. The whole plasmid? Half of it, one strand of it gets transferred over. Yep, you got it. So you got that going on. They also see pili can be used to attach to surfaces, especially if you're pathogenic. So the type 4 pili we just talked about just a few slides back, that's for motility. And so there's different pili. It's usually type something, type 1, type 2, type 3. They also see that, remember back in the transformation process, we said that some gram negatives in particular like to make certain types of pili that they can use to drag DNA into their cell. So pili are another option that many microbes will decide to do under the right conditions. Whoops. So here trying to show pili and fimbriae difference. They're trying to show how the pili are longer. A little blurry. I couldn't get it to crisp up in the focus. Okay. So those are some things that are getting built outside of the cell. All of the flagella, the fimbriae, the pili. This is a slightly different idea, but it's still external to the cell. Here we have what we call glycocalyx. Glyco is referring to sugar. So when we say glycocalyx, there's like two versions. You can say it's a capsule, or you can say it's a slime layer. And really the biggest difference I can see is how thick it is. If it's really thin, we tend to say, oh, look at the slime layer around the cell. If it's nice and thick, then we say, oh, look at the capsule. So it's really just kind of how much is produced, how you're going to talk about it. But either way, what it is it's polysaccharides and maybe a little bit of protein, but mostly it's a watery, sugary goo that the cells are actually making, and then they excrete it out, and then they just coat their whole body with the sugary goo. Sometimes it's referred to as an extracellular matrix, if you want to get fancy. I guess sugary goo isn't fancy enough, but it's a little bit more descriptive, I think. So extracellular matrix is often what you see published. So why would you want to cover yourself with a sugary goo? Got to be some kind of advantage to make all that sugar, and then instead of using it, you excrete it. Yes. Yes, that's a huge advantage. So if you have this big, thick, watery, sugary goo around you, that's viscous. It's viscous. And they have shown time and again, when you isolate a pathogen from a human, like in the hospital, and they send it down to the lab for analysis, time and again, it'll have this beautiful thick capsule around it. The same strain isolated from a natural setting, hardly any capsule. And so they started to see that trend, and many bacteria can do this, but it tends to be that if they can do it and they're a pathogen, they usually do it really well, because if the patient takes antibiotics, that antibiotic, it's really hard to diffuse through that really thick, gooey capsule. And so it really does protect them quite well. And so I don't think they'll let us do this anymore, but years ago, I got a bunch of clinical microbes from the health department in Hancock area. And oh my gosh, the capsule production. It was amazing. So it was like, ooh, okay, good aseptic technique is necessary for this, working with these. We don't work with those anymore, but that was for a different project I was working on. So thick. So what you're seeing here, I think you did look at some capsule slides already last week, but the cells usually stain dark. Or no, you did the negative stain. I don't know if you were able to see the capsule. You probably didn't in your things, but here, if you do the stain right, and if you grow the bacteria right, they'll form this nice capsule. So the cell is dark, the capsule is like that light gray halo, and then the background is dark. And so you can see this is quite significant. I mean, that's a huge amount of capsule that's being put out and coating the cell. Look at that, it's huge. And then if you get a bunch of cells all together, what tends to happen is all of their capsule material kind of flows together, and so you can get this really extended capsule around the whole group. Once you get that level of like many cells all doing it at once, then we call it, oh, that's probably not going to show up, a biofilm. I don't know if you can see that, BIO film, biofilm. So when you get a lot of cells together and they're all making capsules, then because it all coalesces with everyone else's, you can get this really nice big sugary goo. Then we start to describe it more like a biofilm. You have experience with biofilm every morning when you wake up and you run your tongue over your teeth. That's hard to say fast. You get that kind of fuzzy, gooey feeling. That's a biofilm. So all night long, when you were sleeping, the microbes were partying in your mouth, and they were excreting out a way to attach to your teeth. And because your mouth is a delightful nutrient source for them. And so they want to stay attached and they want access to your nutrients. And so biofilm every morning when you wake up. Now during the day, if you're drinking and eating frequently, it tends to wash them off your teeth. And so you don't feel it so much. But not during the night because you're sleeping and you're not eating and drinking. So here's some other nice images of capsules. You can see how extensive it can get. Very, very thick. This one's a little bit more modest. Maybe they might say this is a slime layer in figure B. So this idea that especially if you get a lot of cells together and they all make that capsule, then they say, oh, it's a biofilm. So it's sticky. It helps you to attach, right? You can attach, you don't need Fimbrier, just exude out this goo. And because it's sticky, it helps you attach to the teeth of your host or attach to water pipes. Like cities have issues with big biofilm development in water pipes, the city water pipes and sewage. That can be a real problem. And so just attaching to surfaces and then even evading our immune cells. So the macrophages that are trying to engulf bacteria that come into you, if you have this big, thick, sugary goo, it's harder for the macrophage to actually do that and engulf you and take them into their cells. And so it's a real problem. So it's a good way to evade the immune system. And then it's water-based. So you don't dry out as much. If you get into a stretch of space where it's pretty dry in the environment, if you have that sugary goo, you'll resist drying out. And if you dry out to the point of dehydration, severe dehydration, you're going to die. It doesn't matter what organism you are. All living cells, tissues, and organisms need water. So from the microbe's perspective, there's a lot of advantage to having a biofilm. Yes, good point. In fact, a lot of medical microbiologists are studying this, and they're actually using bacteriophages to attack capsules and biofilms. Yeah, and it's working. There's a nice study that they did with Pseudomonas aeruginosa, I think, which is a human Pseudomonas aeruginosa. I think we might work with it once this semester. We also work with Pseudomonas fluorescens. But Pseudomonas is amazing because it can attack humans, it can attack plants, other animals. It's very versatile. But anyways, they're showing that using bacteriophages to kind of release into that area. I can't remember, they're engineering the phages. I can't remember the details, but somehow they're engineered to break down the biofilm. And so they are looking at that, especially if you think about biofilm producers in your lung, where the lung tissue, it can get harder to breathe if there's massive amounts of biofilm buildup in your lung tissue. So serious problems. Okay, so biofilms and glycocalyx, I'll come back to biofilms a little bit more later. Endospores. So I think it's tomorrow. You're going to do an endospore stain in lab, I believe. And so endospores, I have a love-hate relationship with endospores. I love the biology of it because endospores are absolutely amazing. I hate them because they messed up a good portion of my graduate education, so my graduate research. So endo, within, these are spores that will form inside the so-called mother cell, the bacterial cell. And so they build this endospore within themselves. And it takes a while, but eventually that endospore gets mature and ready to be released. And then the mother cell will, or the vegetative cell will break open and release the endospore. This endospore is amazing because it is incredibly resistant to all kinds of adverse conditions. It can persist for thousands upon hundreds of thousands of years. They've recovered endospores in amber, for example, and then they were able to get it to germinate and grow into a living cell, hundreds of thousands of years later. They even did, was it an endospore that? I think it was an endospore from amber, fossilized amber, that they grew up and they added it into beer production. It was a strain that gave a certain flavor to the beer. And so their claim to fame was we used some bacteria that were, I can't remember how old, super old, coming from an endospore. So I thought that was a great marketing skill to do that. But anyways, really, really resistant. They can get in areas of severe drought and the spore stays stable. Hit it with radiation, it stays stable. Heat, yep, no problem. Pressure, yeah, no problem. Even attacking it with all kinds of different enzymes to try to get it to degrade and break down, nope. So an endospore is a way for that species to go dormant and just survive tough times, extreme tough times, and they just scoff at it and they're like, yeah, whatever. We actually design, not me personally, but the people who build autoclaves, they actually design it to kill endospores. And that's how they decide if their autoclave is actually functioning correctly. And so we know that endospores, you have to hit it with 121 degrees Celsius and 15 pounds per square inch of pressure. And if you can do that long enough, it usually takes a little bit of time, then you can get the endospore to die off. Yes. You can autoclave and kill off endospores, but you have to really do it. Just ordinary, like endospores out in the natural world, it could survive drought, no problem. Radiation, whether it's ultraviolet radiation or even, gamma rays, things like that. Yep, no problem. I mean, they're just amazing. The reason I hate endospores is that when I was a PhD student, there's a master's student in my same lab, research lab, and he was working with bacillus, which form endospores. And I wasn't working with that. I was working with streptomyces, but we used common equipment. And the endospores from his bacillus species got into the common equipment. And so my experiments kept coming up contaminated. The bacillus species take like a day to grow. My streptomyces take 5 to 7 days to grow. So it was so easy for my stuff to get contaminated that I couldn't figure out where it was coming from. And so I did everything I could think of. Finally, we had a vacuum pump that was contaminated. So we got all that cleaned up. Okay, good. No more contamination, right? No more contamination. So I tracked it all down. Endospores were floating around in my beaker of ethanol that I use, with the glass spreaders, you put your glass spreaders in the beaker of ethanol and then you flame it. They were floating around in that. We had a shared beaker that we used to disinfect glass spreaders and things like that. And I just, for grins, I'm like, I'll just take a little bit of this and plate it out and see what happens. And it grew up. bacillus. Like, are you kidding me? So they were just floating around in the ethanol and happy as clams.
Can like any type of spore become in this spore?
No.
So it's only specific.
But that's a good point. There are other spores out there that, like my streptomyces that I like to work with, they form spores, but they're not endospores. This is a very particular type of spore. So let me show you a little bit of the visual here. So here's your vegetative cell or the mother cell that's like, oh, I think I'm gonna start forming endospores. And so endo is within, it's all done within the cell. And then once that's all accomplished and the spore is mature, then the mother cell will break open. And so far, we know that the Bacillus genus and the Clostridium genus, and there's roughly 20 other gram-positive. We have yet to discover a gram-negative that can do this. So for some reason, it seems the gram-positives have this skill set, and the gram-negatives are slow on the uptake and haven't figured it out. These things that I have here, exosporium. So if you look at an endospore and you look at the very outside surface, they call that the exosporium. That's the outside coating. It's just protein. And then there's usually several layers of so-called spore coats beneath that. And then there's a cortex region, which is like the peptidoglycan of the gram-positive. And then right dead center, they call it the core region. That's where you actually have like the cell that is going dormant, right? So basically, it's a cell that has all these extra layers built around it. And then the other key thing that has to happen is that the cell and everything, the whole thing, has to dehydrate. It has to lose a bunch of water and dehydrate. And that's one of the reasons why it's so tough, is that it's capable of dehydrating really low water content. So here under the microscope, you'll see, hopefully tomorrow when you look, you're going to see some vegetative cells that are rod shaped. And then hopefully you'll see, well, hopefully not that. So this bright patch, those are the endospores inside the vegetative cell. Now here you see the spore is toward the end of the rod. So you can see here, the whole rod shape is kind of long. So some species will form their spores right at the end of the rod. Some will do just not quite the end, but just short of it. Some will be more in the middle. I think I have a picture here. Yeah. So terminal spores are right at the end. Club shaped, they often refer to them, and then subterminal, not quite to the end, but they're close, and then more of a central spore. And so you can actually identify pretty well what you're looking at if you have a little bit extra information. Like if you say this was taken from a patient displaying these symptoms, and then you look and see this, then you go, oh, and you call the CDC, because some of these are like Clostridium botulinum. I think this one might be Clostridium. And so, they can often diagnose just by position of the spore with the context of how sick is this person, what are their symptoms. We're not working with anything dangerous tomorrow. We're working with normal bacteria. It'll dehydrate inside all those layers, the exosporium and all the spore coats. It's dehydrating in there, and then it can stay stable for thousands of years.
Okay.
Yeah.
So that's not what it came like.
So then to reverse all that, it means hydration. You got it. And so that's what you do if you want to get endospores up and running. You pop them in water or a saline solution. So let me just show you this. I'm not going into the details. I'm not expecting you to know step by step. I put this here more to show you they've done a ton of work on this, because there are some really deadly pathogens that do this. And the fact that endospores are so tough and can live for so long you can see that it's a step-by-step process. One thing that they've discovered in the lab as they're playing around with this and running experiments, once the cell decides I'm going to form endospores and they kick into the phases, and then if you like give them a flush of nutrients, like you don't need to do that. Here's all these lovely nutrients. Once they start, they can't stop. Like they can't back out and go, oh, never mind. I'm gonna just stay normal and not do my endospore. So it's a commitment. You know, they're like, they say it's a commitment. If they're gonna make an endospore, once they kick into it, you gotta finish it. And I think with endospores, I think it's an 8 hour in the lab when they do the process. I wrote it down. I think it's like 8 hours. Yeah. 8 hours from start to finish in a lab environment. So it's a commitment. And then they've mapped out 200 genes that are necessary to go through this whole process. Yes.
What happens after next time it pops off, then the rest of it dies?
Yes, the mother cell will die. Yes, yeah. But then inside that endospore, all those coats deep in the core, there is a cell there. It's just super dehydrated and it's just waiting, biding its time. And it will get active again when the conditions are right. So it's a way for them to survive tough times. But if times become nice, though, that's endospore, we call it, it'll germinate. We actually use that word, it'll germinate. Here's another process here of trying to show the mother cell or vegetative cell going through the stages here. And then here they're showing some of the dehydration chemistry going on. So I think I have it in one of the, so some of the dehydration events they make during those steps, they'll make this special chemical called dipicolinic acid, which helps. It's a way to kind of protect the DNA and shelter it from any damage during the dehydration process. And SASPs, they call them, small acid soluble proteins, or they'll say SAPs. These are, again, to protect. And so as that desiccation is happening, they'll make these special chemicals. It also can protect them against high heat. Like I said, you really have to autoclave endospores for a long time if you want to kill them. So that's actually, companies will sell you a vial of endospores that you can pop into autoclaves to test your autoclaves. Unfortunately, they're super expensive. It's ridiculous how expensive they are. It's like, I mean, we could just grow up our own endospores and use that. I mean, and then they sell them in cases. I only need a little bit, but they're like, no, you got to buy the whole case. And it's, I think it was over $500 just for that. It's like, yeah, I know how to make endospores. I can do that. So here you see it germinating. So there's your endospore in figure A. Unless you do a very special stain, it'll pop that kind of, it'll reflect light off of the outside of it. So it looks really bright to our eyes under the microscope. And now you see it's starting to rehydrate and start to activate, and now it's starting to germinate. And it'll get back to its original size as the mother cell was, the vegetative cell, and live its life. Okay. And here's kind of a electron microscope view. So you're going to do an endospore stain tomorrow. Endospores are really tough. All those spore coats I told you about, the exosporium, takes a lot to get that stain to stick. And so we're going to use heat, high heat, boiling water bath. And we're going to soak malachite green. We're going to use malachite green, and we're going to use heat to push it through the cell and onto the developing endospore. And so you're going to be looking for that kind of color. They'll explain it to you more in lab. So hopefully we'll see some in the cells and some free floating, and you'll get a view of endospores. Not dangerous ones though. So, all right, we'll stop there and meet up again on Wednesday.
Oh, dipicolinic acid.